All right, well, I'm super thrilled to have our guest, Chris Gampe, here today to tell us about strategies to extend duration of action for oral and inhaled drugs. So Chris is a principal scientist and team leader in the discovery chemistry group at Genentech in South San Francisco, a rising star there. Previously, he was at Novartis in Cambridge, and he did his education in Germany, receiving his diploma in chemistry from Wurzburg, where he tells us X-rays were discovered, and got his Ph.D. from Zurich under Eric Carrera, completing a natural product synthesis, and did his postdoc with Dan Kahn at Harvard in chemical biology and microbiology. So before we get into this discussion, I do just have a few minor announcements. First is I do want to thank our sponsor who's making this webinar free. iPRECIO is a company that provides innovative drug infusion technologies for lab animals. They create these cool, controllable, implantable, programmable, and refillable pumps for animal studies. This is actually a tool that I was asked about in my Genentech interview, so that was pretty cool to get in touch with them. These mini pumps, what they help do is administer doses at controlled programmed levels. So if you have a compound that doesn't have perfect PK, like we're going to talk about today, you can use this to mechanically administer a dose and control the levels of drug in an animal. This can be super helpful in vivo pharmacology when you don't have a perfect tool compound with great oral exposure yet, but you do just want to see what the on-target toxic effects or if there's any efficacy at a baseline. So we have a recent article on this called Minimum Viable Experiments with Imperfect Molecules, a mini pump mini review. And if you search Drug Hunter for mini pump mini review, you should be able to find that article. People are probably familiar, or at least the pharmacologists in the audience are probably familiar with Alzet osmotic pumps. These are, just based on osmosis. You implant them and they sort of slow release drug. These pumps are have a chip in them and they're programmable so that you can control the dosing over time and you can refill them so your pharmacology teammates don't have to, go in and adjust the animals, every day or something like that, especially if you have a long study for something like oncology. All right, and stay tuned. In 2 weeks, we're going to have Hande Ozdinler and Professor Rick Silverman with us to tell us about Rick Silverman's next big drug for upper motor neuron diseases. Rick Silverman was the inventor of Lyrica, and so it'll be great to see his team from Northwestern tell us about this new topic. All right, so I just want to let Chris tell us a little bit about himself. So Chris, what is your favorite drug? What inspires you in drug discovery?
Okay, well, so, being born and raised in Bavaria, I would have to say personally, my favorite drug would be recreational, namely beer, with the API being ethanol. But all joking aside, this is a really difficult question. Every time I'm impressed when I see something working, a drug working in patients and changing people's lives, from something as simple as giving your teething 9-month-old a dose of an NSAID at 3 a.m. and buying everyone in the family another 5 hours of sleep. And of course, it's on one end of the spectrum, but there's so many life-changing and disease-changing drugs out there. It's really hard to pick a favorite. If I had to pick one, I just randomly, the first thing that comes to mind would be the voxelotor drug. I think you had a post on that too. I just think this is an incredible story. It's a covalent drug, right? That modifies one of the sort of quintessential life-giving proteins, which is hemoglobin, and it partitions into the red blood cell, and it covalently modifies that to allosterically change the oxygen affinity. And I mean, from a drug discovery standpoint, the audacity to bring this through and believe in this mode of action and carry it forward is quite impressive. The mode of action scientifically is super interesting. The backstory, if you ever get a chance, is a story certainly of failure and perseverance, and it's disease changing, right, and truly modifying for patients. So I think that's a really cool drug.
Yeah, good choice. Timely too, because I saw Global Blood just got acquired by Pfizer, right? So
Yes, that's right. Yeah.
interesting, yeah, point in that story. And you and Vishal actually have a review on aldehyde drugs, right? Was that you
That
and
That's right. Yeah. So this was, I guess this is kind of why we got into it, looking into, we had a project where we had some aldehyde hits and needed to refresh our memory on what is actually known about aldehydes, because everyone looks at an aldehyde and it just kind of throws up their hands, goes, nah, this is never going to be a drug. But we wanted to put some I guess meat on the bone there and do some literature research to find out, what aldehyde drugs are out there and how, if you were to go into discover and develop one, how would you do it and what are the pitfalls? And so the Voxeltor was a really great case study there, but there are plenty others out there too.
Cool. Well, yeah, beyond a specific drug, what do you think is the most exciting area of drug discovery to you personally today?
I mean, everyone talks about AI, right? I think it is really cool. I'm very much looking forward to seeing how it's going to impact synthetic chemistry with retrosynthesis designs, and free up medicinal chemists to think even more about the quality of their designs and less about how we would make a certain molecule. It's already impressive to see how it impacts structural biology with AlphaFold and just being able to predict protein structures out of thin air, seemingly. And I'll be very interested too to see how it impacts the microbiome field. So this is something that I've gotten into mainly out of curiosity, right? A lot of us have, or I guess there's been this long-standing of knowledge, semi-knowledge, well, the gut microbiome somehow influences health. And over the past 10 years, there's so much more that's been discovered there. A lot of it is still correlation, right? The composition of your gut microbiome changes in disease, but does it change as a consequence of disease, or is it causal for disease? And any kind of disease, not just inflammatory bowel, CNS, even immuno-oncology. And so just deciphering what it is in the microbiome that is actual causal for disease modulation or therapeutic outcome will be super interesting. And what you have to do is essentially, or what I'm mostly interested there is the chemical aspect, right? Metabolites that the bacteria generate and how they interact with the host. And I'm hoping that AI can help, and machine learning can help with the unbiased metabolomics that you need to do to actually figure out what are the compounds that are being generated there by the microbiome that change host biology. So yeah, this is kind of what I'm excited about right now.
Yeah, very cool. Yeah, I think the microbiome is super underappreciated. I remember when BMS bought Celgene, where they had that 1 or 2 year hold on their approved drug, that $1 billion drug, because of the bacterial metabolism of the drug. So
yeah, of course, that's another aspect, right? But then also, I mean, the fact that if you get certain immuno-oncology drugs and the efficacy depends on your microbiome composition. I mean, it's just mind blowing.
Yeah, it's crazy. All right, so last question, and we have a lot of early career scientists in the audience as well. If you could go back in time to when you first started drug discovery, what advice would you give yourself?
I would probably, I think what I personally struggled with most early on in my career was moving from a setting where you're somewhat autonomously working in lab, right? So my background is in total synthesis, and then I did some chemical biology. And there are factors, of course, that influence your work that are outside of your control, but they're mainly scientific. and you know how to respond to it by yourself oftentimes, or you troubleshoot your synthesis, and you have the autonomy to just walk in the lab and just do it, right? Nobody's going to dictate what you're going to do, or tell, or even ask you questions in that moment when you set up that reaction, because you're the world's expert in that. When when I moved into industry, I think what I struggled with a little bit was adjusting to an environment where you are working within a huge group of people, and so many things are outside of your realm of influence. Decisions are being made, strategically, um, based on, totally outside, well, above your pay grade and outside of your realm of influence, but it impacts your daily life. And I think what I had to learn more or less the hard way over the years is to focus really on what you can influence, and to not get derailed by things that are just beyond your control. It's easy to just kind of pound your head against the wall and be like, yeah, but this shouldn't have been this way, or why was this done, and just get so frustrated that you become disabled in your work life. And finding ways to just accepting some of these things. I mean, okay, I'm going to change what I can change, and I'm going to work on those things. Was just that mindset, I think, in some ways was a game changer for me. And then you become much more effective in your position, and in interacting and influencing beyond the reaction that you can set up yourself, right? And you still retain the autonomy, but yeah. So I think focus on what you can personally influence, and don't get too hung up about things that are just outside of your control.
Yeah, definitely. Super helpful. And one of the 7 habits of highly effective people. So fantastic.
Yeah, just maybe if we have the time, just a brief anecdote there. I remember writing my first paper that came out of my industry efforts, and I was very invested in it. Right, it was my idea. I was super excited about writing this paper, but of course there were co-authors, right? People that had influenced it one way or the other. And I remember probably weeks long discussions around how to write the introduction, and I felt very strongly about it. But then I realized, okay, no, there are other people in the field that have different opinions, and what they kind of contradict what I'm saying. What am I going to do? And I got really hung up on it. And it took me a while, and it actually took my former boss to say, okay, take a step back and see what is it, what's the outcome that you're looking for. Like, well, publishing this paper. Okay, what do we need to do to get there? Well, maybe jump over my own shadow, and not be so stubborn, and just make it happen, and focusing on that. And so that was, yeah, that was a somewhat of an influential episode there from.
Yeah, I'm sure plenty of people in the audience can relate. So yeah, well, thanks so much for sharing your thoughts. I'm going to turn it over to you now so you can share your slides and
All right.
yeah, tell us about the main topic.
Okay.
Thanks, Chris.
Cool. You bet. 1 second. I'm going to ask the obligatory question if you can see this.
Yes.
Okay, great. So, the way I got into this, so I guess first of all, I should say that this is going to be more of a teaser or a discussion background to start a discussion, because it's a very I could have talked a lot longer on this. And I will focus mainly on strategies as they pertain to medicinal chemistry. There are a lot of strategies focused on formulation as well. I'm not an expert in that area, and so I'm going to focus primarily on the medchem strategies. But the way I got into this, and why I got interested in this at first, was a few years ago, where we had sort of tried a lot of things to decrease potency of a certain molecule, make it more potent. And our human dose projection looked quite abysmal, many grams per day. And so we had to look for other ways to reduce the dose. But this kind of goes to also show that in my experience, I think oftentimes teams focus primarily on in vitro potency first. And there are various reasons for that. And of course, potency is important, but I think sometimes potency is being used as a synonym for human dose, right? Because that's what ultimately matters, human dose. And the belief is, well, if we just make it more potent, it can never hurt, and it's going to be leading to a lower dose. Well, back then when I started looking into this, I came across this paper by Gleason and coworkers, and I thought this was really cool. So they went through the hard work to analyze 261 oral drugs and plotted their mean therapeutic dose here on the y-axis in the clinic. So this would really ultimately matter, so our dose, versus their in vitro potencies, XC50 here, because it's either an IC50 or an EC50. So essentially correlating the very beginning of your flow chart, primary cell-based or biochemical potency, with the very end of your flow chart, which is human drug. And you can see here the correlation is rather poor. So if you were to only dose predict or do the mental exercise of predicting a dose based on primary potency only, you would be more often wrong than right by quite a bit. And I thought that was pretty interesting to drive this home. And so, of course, people now would say, well, duh, you're ignoring all the PK properties, right, of this molecule. And of course, that's what has the big influence on dose. And so, when we're now talking about duration of action for oral drugs, plasma half-life, as you can read it out in a PK experiment, often correlates well with duration of action. And so there are cases where that's not the case. But if you have to inhibit, let's say, inhibit an enzyme, you know your minimal concentration that you need for efficacy, and your drug plasma concentration needs to be above this threshold. And so once it dips below, your duration of action is gone. And so in this case here, you would have what this be 18 hours or so duration of action. And you can see that half-life, as you defined by going from Cmax to 50% Cmax, and the time that it takes to go there, is correlated well with duration of action. Half-life can be read out in an IV PK experiment, and there are two very critical parameters, volume of distribution and plasma clearance, that influence and dictate half-life. And this is a really cool paper here by Pfizer colleagues that I can point to you. And so when we looked for other ways of lowering the dose back then in this scenario, we came across this paper in 2018 by Merck colleagues. And they show these really two cool plots here. So this is human dose on the y-axis versus the half-life, human PK half-life, right? And for a given molecule with a given potency, and you can see that if you go from human half-life of 2.5 hours or so right here to a human half-life of 8 hours, you would likely decrease your dose by at least tenfold without touching potency at all. Same potency, just different half-life. Of course, this is hard to read out, half-life in humans, but you can allometrically scale that back to rats, which is what we more routinely can read out, and you can correlate human dose with the rat half-life. And so if you're looking at compounds with a half-life of half an hour, you were able to extend half-life and duration of action in that respect to 4 hours, you'd get 100-fold lower dose right there without touching potency at all. And so the real take-home message, of course, is plasma half-life is an important and optimizable parameter to lower dose. But how do you do it in a metchem team? Well, a lot of times I think first go-to is lipophilicity. Let's bring down lipophilicity. It's going to change, it's going to reduce clearance, and that's probably a good thing. Well, it turns out that if you change log D, or if you reduce log D and therefore clearance alone, that oftentimes doesn't work to extend half-life. And so this is a paper from Fabio Porcatelli, your former colleague from Genentech, where he analyzed for almost 5,000 compounds, non-charged compounds. So these are neutral compounds, and plotted the clearance here on the y-axis, rat clearance versus volume of distribution. And the shading here correlates with the log D. So the darker spots are compounds with higher log D, and the lighter spots are compounds with lower log D. Meaning that as you reduce your log D, you go from a space up here to down here, because there is this really nice correlation, right, between these two. And the reason is because log D influences clearance, right? As you go down here in log D, you get to lower clearance, but it often also tracks with volume of distribution. So you reduce your volume at the same time as you reduce your clearance. And so overall, rat IV half-life, as shown here on the y-axis, doesn't change dramatically if you only look at log D. So these are these box and whisker plots here, where you can see in the usual log D space where we're working oftentimes between 1 and 4, there's not a dramatic change in IV half-life oftentimes. So log D alone, changing log D alone will likely not extend half-life. So how do you do it? Well, if you look at the math, this equation for what dictates half-life, again, it's volume of distribution over clearance. So you have to modulate those two values independently. Right? You can't decrease clearance by tenfold if volume of distribution decreases by tenfold either. And so this is, again, data from Fabio's paper, where he looks at the number of matched molecular pairs in which half-life was extended by at least twofold. And so we just talked about this, right? This half-life only reducing log D will not most likely not do the job here. Reducing clearance is a better strategy, right? You'll be successful some 70% or so of the time. But if you can reduce clearance and increase log D, which is really sort of counterintuitive, this seems to be the most promising strategy, as opposed to reducing clearance while also reducing log D. So then the question, of course, is, okay, well, great, but how do I do that? And Fabio and the Merck team here looked again into their internal databases and came up with some recommendations, matched molecular pairs here, transformations that were half-life efficient, essentially. And the bottom line is you need to introduce what's been termed non-metabolizable grease. So if you take a look at the left-hand side here, this table, right, these transformations, nitrile to fluoride or proton to CF3 group, most of the time you will increase the log D. So the delta log D is positive here most of the time, which leads to, on average, also an increase in volume of distribution. But it does not lead to, it doesn't change clearance as much, right? So the change in clearance is oftentimes here actually reduced. And so obviously this is a really half-life efficient transformation. If you can increase log D, increase volume of distribution, and decrease clearance, this is a great way to extend half-life. The Merck colleagues did a similar exercise here, highlighting these half-life efficient transformations with a little blue star, and the same things popped up, right? Introducing halides here, oftentimes as non-metabolizable grease, will move volume of distribution to a greater extent that it does move clearance, and therefore that ratio will increase, which is what dictates half-life. And the opposite is true too, right? If you're introducing essentially metabolic hotspots, or like an hydroxy potentially, or a carboxylate, which will most likely reduce your volume of distribution, right? These are half-life inefficient transformations where you probably suffer in terms of half-life. There's one other figure here in the Merck paper that I wanted to point out, which was, again, really interesting. They dug out fragments that they observed in compounds with long half-life, and those fragments are shown here in blue, and compounds or fragments that were primarily observed in compounds with shorter half-life. And so you'll see that primary amines or amines in general pop up here. Reason is most likely because basic amines have a much larger volume of distribution, relatively low log D, so you're probably going to be relatively half-life efficient there. Of course, sometimes you just can't tolerate an amine in your scaffold, right? So you need to, of course, make sure that this is tolerated from an SAR standpoint and potency standpoint. But there are a number of other interesting, I thought, heterocycles here that they dug out. And again, on the opposite end, right, introducing more acidic groups here or metabolizable polar groups is probably going to hurt you. Okay, so this really in a nutshell, and I'm going to switch gears now to talk about the inhaled drugs. And I know that this might be, or this is probably more of a niche knowledge or niche field. I mean, inhaled drugs are hugely influential and very important, and there are quite a lot of efforts to come up with more inhaled drugs because of the inherent safety considerations that are pretty positive, where you dose locally to the lung and you minimize systemic exposures if the lung is the diseased organ. So we started looking into that on a project. You know, how do you extend duration of action for an inhaled drug? When you dose through inhalation, meaning topically into the airways, right, primarily on top of the epithelial cell lines. If you go to the literature, there are really 4 validated strategies to increase duration of action in the lung. One of them is through low solubility or dissolution rate, because the drug is going to be deposited up here and slowly going to dissolve. The same thing, the next one is low permeability, right? You want to keep your compound out of permeating into this first layer of cells. And then there is pKa, which seems to play a role by trapping compounds in either the lysosome or sort of non-specific binding. And then the fourth one would be through long receptor residence time. Although, in my personal opinion, I think there are some caveats here. But I'm going to show you some data that supports these claims that I just made. So this is lung half-life, permeation half-life in minutes here versus permeability readout. And you can see the lower the permeability, the longer the half-life in lung. This is a similar graph for dissolution rate and solubility. So the lower the solubility, or the longer the dissolution rate, the higher the half-life. And then here's a graph that plots lung PK for monobasic compounds here versus a dibasic compound, clearly illustrating, it's one example, but there are many more of those, clearly illustrating how basicity influences the ability of the compound to stick around in lung. And probably the poster child example for the long residence time here that you can optimize is the switch from ipratropium to tiotropium, muscarinic receptor antagonists, where there's a nice correlation of the on-target sort of dissociation half-life on the receptor, going from relatively short to longer to dose, right? This is QID, that's 4 times a day, 40 milligrams. This is once a day, 18 milligrams, so quite a reduction in dose. Now, big caveat here, and I have it here. As you note, these are PK parameters, right? We're looking at half-life. Now, this does not necessarily track with duration of action, and the reason is that it depends on where your target is. And these strategies have been validated mostly for targets that are extracellular. Muscarinic acid receptor is an example here. And you can appreciate how if you deposit lung on the cell surface here, even if this compound has low solubility, very low permeability, you might still be able to engage your target. If your target is intracellular, you're going to have to permeate into the cell, or diffuse into the cell, engage the target here, but then avoid the compound being diffusing quickly into systemic circulation and clearing it. So this is the delicate balance that you need to do. And there's one key, or there are a few examples, but this is a very influential example to me, at least, that I looked at, that we found when I teamed up with Karen Sammy in our department to look at some of these strategies. Big caveat again, lung retention as a PK parameter for inhaled compounds is necessary, but it's not sufficient for duration of action, because you can measure the compound being around, but it might just not be able to engage your target. And this is an example here, PI3K inhibitor from GSK. They took this compound into the clinic. It has very low solubility, and they measured lung exposure for 24 hours in humans. Okay, so this is human data, but they saw that target engagement dropped off after 3 hours. And this is likely because PI3K is an intracellular target. So if you deposit the compound out here, you can measure it in a PK experiment, but it won't be available to engage your target. So big caveat here. So when Karen and I teamed up to take a look at, how do you actually then do it, right? Especially for intracellular targets. At the end of the day, I think our recommendation is there is no blanket statement, how you would do it. The way we recommend doing it is you let your in vivo work guide what the MedChem team has to optimize. And the way to do it is, in our opinion, is you have to have access to a relatively high throughput PK/PD model where you can measure a PK/PD time course, because the time course shows you your duration of action, and you read out lung, plasma exposures, and target engagement, so you can assess if there is a disconnect between PK/PD. And importantly, you dose in solution. And I think that's important, because you take out any variables associated with insolubility or dissolution rate of the compound, because you want to really find out what are intrinsic molecular features that drive lung retention. And so then if you have this model, you can look into your database or in your chemical series, and you pick compounds with a diverse set of in vitro properties. And this is really up to you. I just threw out a few here: basicity, in vivo half-life, MDCK, whatever you can come up with, a diverse feature, and then you look at how that impacts PK/PD, and you start establishing this correlation between in vitro and in vivo duration of action. And there are 3 papers that I'm highlighting here that have done exactly this, and this is what we base this recommendation on. And as you can tell, they came up with quite different recommendations. In this one case here, it was primarily low permeability that drove lung retention. And then, of course, low permeability as a factor of molecular features such as high molecular weight, high PSA, high hydrogen bond donor numbers. Then there was a different paper that talked about, wait a minute, we actually need kind of low log D compounds, but we also found out that the IV half-life needs to be relatively long. And if we combine these features, we observe long duration of action for our inhaled compound. And the third one is sort of a hybrid. They again found out that volume of distribution seems to drive some of this log D, pKa, basicity, but also permeability. And so, our recommendation is really, this might be target specific and series specific. So I think you need to have access to an assay cascade like this to really define what you need in the specific circumstance. And I think with that, I'm just going to open it up to questions. I mean, as I said, again, a lot of stuff in here, couldn't cover it comprehensively. We could have talked a lot longer, but I hope that we can use this as a jumping off point for further discussion.
Awesome. Thanks so much, Chris. I actually got a text from somebody in the audience saying, I have been trying to convince my boss that T-half is more important than potency for our backup program. Haha, this talk is gold. So sounds like you've already made an influence on somebody here.
Great. That's great to hear. I was in a similar boat, except I needed to influence the team, but it doesn't matter. Whoever you need to influence, I hope it helped.
Yeah, definitely have to save this one in everybody's notes. We have one question from Sam Munn. He says, do transporters play a role in lung PK?
So the current thinking is that transporters oftentimes or will unlikely play a role in lung PK, and the reason for that is I guess twofold. First, there is some data suggesting that there are the expression levels of the notorious exporter pumps are not extraordinarily high in the lung epithelial, but probably more importantly, it's a topical administration, right? So you're going to have a very high local concentration and concentration gradient across that first epithelial cell membrane. And that's likely, even if your compound were a transport substrate, and even if this transporter were expressed highly in the lung epithelium, it would likely be overwhelmed by just the local concentration of your drug. So the general notion at this point is that it's not a very important feature to look for.
Gotcha. I have a question about your, before we go on to more general questions, the example you showed with the tiotropium, the longer half-life, I think it went from like, 20 minutes or something to 35 hours.
Yeah.
I think it was the target residence time that was
That's
changing. Right. Yeah, I guess, do you know what the mechanism is for that increased residence time?
I mean, I would have to say that it likely is driven by the off rate, right? I mean, it's just a different way of saying half-life. And it came from just meticulous metchem optimization. I will say though, my personal, or let's put it this way, it's really hard to establish that this, again, very first line assay of, receptor occupancy, oftentimes maybe with an SPR assay or so identified, something very reductionist sort of on the front end, how that correlates with dose, because there's so many factors that you influence. And if you go, as you change this receptor occupancy time in your, let's say, SPR assay. For example, we found a few cases where teams set out to use that strategy and said, we're going to set up an SPR assay and we're going to relentlessly optimize sort of off-rate receptor target engagement or time half-life for target engagement. And then we're just going to, hope for the best. And what oftentimes happens is that compounds are being optimized into a very high log D space, because sheer lipophilicity will drive potency, will change your off rate, and you will end up with a compound that just sticks to your target for a long time. But it also, of course, influences permeability, solubility, and everything else. And so then if somebody doses a compound by inhalation with a log D of C log D of 6 or so, right? And then they say, well, listen, this has this long half-life on the receptor, therefore it's going to have long duration of action. I would always say, well, it also has a very poor permeability, so, very poor solubility. So you're going to likely see long residence time in the lung, but I'm not convinced that it's because of the on-target residence time. It might just be sort of the insolubility. So there's so many factors at play. I would take that strategy with a huge grain of salt or caveat there.
That makes sense. So we have another comment from Rao. He says, nice talk, Chris, thank you. And he says, increasing the volume of distribution will increase the distribution of the compound and hence the off-site effects. Any thoughts on that?
It's always a concern, for sure. I will say that, and that's something that Smith points out in his paper that I cited, Pfizer colleagues, that yes, larger volumes of distributions, what they tell you is that where the compound is not, and that's in plasma, but you do not know where exactly it is in the body. So it doesn't, it doesn't tell you much where in the tissue it is. But of course, as you point out, there are some concerns with that. That's why people usually shy away from, highly lipophilic basic amines with huge volumes of distribution. So as always in medicinal chemistry, right, there's not one strategy that you can drive to the extreme and you're going to be successful. But I would say in this, to modulate half-life, you have those two parameters, clearance and volume. So yes, you can drive up clearance and that will change your half-life if you do not change clearance. But you don't have to drive it up as much if you can decrease clearance at the same time, right? So you probably can maintain a pretty reasonable volume of distribution and just change, the ratio of those two values. So
Yeah, makes sense. All right, we have one last general question. They said, would you recommend people with organic chemistry backgrounds who want to get into drug design to do a postdoc in chemical biology or biochemistry to broaden their scientific horizons? Obviously, you've been very successful with postdoc in chemical biology. What are your thoughts?
It's difficult to give this general advice. My general, even maybe more general advice would be follow your passion. Do what you really want to work on, or work on what you really want to work on, because ultimately, this will drive your daily motivation to get to work and to get stuff done in lab, which will impact your impact in lab and your success and your ability to tell a great story at some point at an interview, saying that, no, these were the reasons why I wanted to really study this certain aspect of science, and look what I found. And sort of just this passion, I think, is much more important than strategizing around, what is the best postdoc to do to get an industry job. If your passion is with photocatalysis or whatever, solar panels, right, then go for it, by all means. I think you'll be successful and you'll have a great story to tell. If your passion is chemical biology, great. If you want to do another postdoc in total synthesis, if your PhD is in total synthesis and you have the motivation to do it and the reasons to do it, go for it as well. I have to say, so this would be my general advice, not knowing anyone's background, right, specifically. For me, I think that was my personal driver. I wanted to learn more about how small molecules that I, by that point, had, gathered some reasonable education on how to make small molecules, how those small molecules impact and influence biological systems, and finding ways of how to study this influence. And so that's what drove me to, that was my motivator to do a more chemical biology postdoc. And I have to say, it has served me well, I believe, in my career, because it just, I've expressed proteins in my postdoc. I co-crystallized them with small molecules. I went to a synchrotron. It was a lot of fun. I've not done that in my industrial career, but just having gone through this, I think, builds awareness of what are sort of the caveats that the other disciplines face. What are how do they think about problems? And it just builds sort of a common language to talk with biology colleagues, be it structural biology or chemical biology, or you name it. But I think it just broadens, I think, your view a little bit. That said, I don't think it's critical. This is something you can learn on the job too. And that brings me back to, I think, just if you can, I would advise anyone to just follow your passion.
Yeah, fantastic. Yeah, and talking to biology colleagues is certainly probably half the job at this point. But yeah, okay. So people are all grateful for your talk. I'm very happy to have had you as a guest. Thank you so much. And yeah, with that, I think we'll close the session.
You bet. It was great. Thank you for having me. It was a lot of fun. I thank you all for attending.
Thanks so much, Chris. Talk to you again soon.